Good afternoon, everyone. First, I want to thank Genomics Bioscience for organizing such a wonderful event. I want to take this opportunity to share with you some of our recent development at Sage Science. So, outline my talk. Why do we care long-range sequencing? I'll briefly talk about that, and then I will introduce briefly about Sage Science and what we do at Sage Science. I'll spend most of my time introducing you a new platform called Sage HRS, as well as a new tool for capturing large specific genomic fragments named Catch. And along with that, I will introduce you a little bit about the results about using long-range sequencing on Catch products. Well, so Adam talked about this, showed this picture earlier today, and what I want to add to that is at the bottom, I added a couple of data points. Well, of course, that's from the government website. And what I want to point out is even sequencing cost has dramatically reduced over the years. And it seems like it has stabilized in recent years. And it's around $1,000 per genome. It's still probably somewhat expensive. Short-read sequencing has been very dominant in the past decade. Everybody knows about Illumina sequencing. They are really good for a lot of things, high throughput, and very accurate SNP typing. And they are widely used in genetic testing, companion diagnostics, as well as drug development. However, short-read length is not able to reveal all the information, particularly with regard to complex genomic regions due to structurally variable nature of the genome. It oftentimes makes it very difficult to do de novo assembly of some genomes. If we talk about like plant genomes, polyploidy, and all the stuff, it makes it very difficult when using a short-read sequencing. So this, it's kind of similar to what this picture shows. A lot of genetic information is actually missing from short-read sequencing, and that makes kind of urgent for the field to develop more of the long-read sequencing solutions. In contrast to the short-read sequencing, the long-read offers a number of advantages. And it first eliminates PCR amplification based biases. The long-read can span repetitive sequences in the genome, as you can imagine. It reveals, possibly can reveal structural variation beyond SNPs. For example, large indels, copy number variation, fusion, translocation, and so on. Long-read can also resolve heterozygosity. Populations are always, very highly mixed, and long-read offers a possibility to resolve phased haplotyping. Currently on the market, there are a number of categories for long-range solutions. And the first category is called something called synthetic long reads. The most popular one is perhaps 10x Genomics, which is basically a droplet-based partitioning system that utilizes a technology called linked reads. The second category is some real long-read sequencing, single molecule sequencing. The most known ones are PacBio and Oxford Nanopore. The third category is optical mapping. Basically, in their platform, they stretch single molecules out into, then push them into nanochannels. And those are particularly good for large structure detection and not necessarily good for any small, small things like SNP. Whatever long-range solutions you favor, one of the most challenging issues is sample prep. Because when you start with any of those platforms, you need a high molecular weight DNA. Because DNA, particularly high molecular weight DNA, they can easily break down during extraction stage or any liquid handling. Sequencing rate, according to technology platforms, it's not really limited by their technology, such as Oxford Nanopore. It's rather limited by library preparation process. So at Sage, we are really dedicated to generating high quality, large DNA molecules for just sample prep. So Rob from Lucidgen talk about something ultra small, and I'll be more talking about something ultra large. Over the past number of years, Sage has developed novel products that primarily focus on size selection. And a couple examples. Early on, we developed a pipping prep that really purified and cleaned up the library for short-read sequencing platforms like Illumina, and by removing all of those adapters, primer dimers type of stuff. And blue pipping as has played a really important role for the longer sequencing read space. So to go further a little bit into the detail, and the left figure shows the DNA size after size selection on blue pipping. The right figure generated by PacBio a few years ago that really showed we easily doubled the M50 by using size selection on blue pipping platform. So more recently, we developed a new cassette called a high-pass plus cassette, and that also fits into the blue pipping instrument. And to quickly show you what we got by sequencing the samples after size selection on Oxford Nanopore Menaon instrument, we easily more than doubled the M50 after size selection using the high-pass plus cassettes on blue pipping. Now, I want to talk about a new platform called Sage HRS. That's a new platform we recently launched, and particularly focusing on sample prep for long-range genomics. The input for HRS is intact cells, nuclei, or even blood. The good thing is the enzymatic, DNA extraction and enzymatic modification takes place in one integrated solution operation. Most importantly, high molecular DNA on this platform is protected from being shared during liquid handling process. We have developed 3 major applications on this platform. The first one is high molecular DNA extraction. We can extract DNA really large, up to 2 megabases, and I'll briefly talk about the data. And I will spend most of my time talking about catch, that we used that on the HL system to capture large genomic fragments of interest and using the CRISPR-Cas9 technology. And I won't talk about the third application today because of the time. Here is the cassette that we put into the instrument. Briefly, the top is something called a reagent well, and that is a sample well where you load the cells in, and the DNA migrates along the agarose gel column. At the end, you can elute the DNA to the elution modules. You collect them for downstream applications. This cartoon shows how the process is conducted. And again, you load the cells to the cell wall. And when you start electrophoresis, the SDS reagent in the lysis buffer moves along and lyses the cells. And then the electrophoresis will drive the SDS or any other junky stuff through the agarose wall and push them over, leaving intact, gigantic chromosome level of DNA on the face of the sample wall. They can be well above 10 megabases. Because the DNA is so big, they're not able to move into the agarose gel. After that, you can apply, depending on your needs, you can do enzymatic reaction. If you want to do whole genome fragmentation, you can simply use NEB's fragmentase to break down the DNA. And depending on your needs, you adjust the amount of fragmentase you use depending on what size of genome fragmentation you want to do. And you can also do Cas9 nucleus that targets a specific region you are interested. So isolate that element alone and then move along through the agarose and collect them. So this is a post-field gel picture that shows how big the DNA can become after the extraction and the selection on the HRS platform. And we can recover over 50% of the DNA input. So catch was initially coined by a collaborator in Tel Aviv, Yuval Epstein. So it represents Cas9-assisted targeting of chromosome segments. The concept of catch is rather simple. You design guides on both sides of the region of interest. So Cas9 will specifically cut the target, and then you use SageHR's system to take them out. What I want to emphasize here is that using this approach, we can actually do very large elements. We can go 100 KB plus. We can go 200 KB, 400 KB, and all the way. So I'll show you more of the data in a little bit. So like I showed earlier, the sequencing cost seems like have stabilized a little bit in recent years. It's almost like 1,000 genomes, or $1,000 per human genome. So the target sequencing is still very valuable because it's still cheaper. It's probably in terms of time it's faster. And what's more, with the catch approach we apply here, it focus on the large fragments that you are interested. And a lot of the target enrichment approaches out there, they are pretty much based on probe-based hybridization. And with that approach, you cannot really go too long. So IDT's recent target enrichment approach, I mean, they call that long read, but they are really focusing on, or can do all the way to like 5 KB-ish. But here we're really talking about over 100 KB. To give you a refresh of the workflow, sorry, you load the flow, you load the live cells to the sample well, you lyse the cells, and you apply enzymatic reaction, you cut the target, and electrophoresis move the target into a certain distance. If anybody is familiar with Sage as a product, you can always do the programming. You can determine the conditions, and you know what you want, and you know how where the fragment will migrate to depending on the size. And you load the DNA, and you collect that for downstream analysis. After that, you can simply do, even though we know where the DNA will migrate, depending on the size, and we can still do a qPCR validation to really determine where the fragment goes. And also, we can use the qPCR to determine how much DNA we recover from the initial input. And like this case, it's a 200 KB mouse BRCA1 fragment, and it was enriched in elution module 4. And the enrichment is well above 200-fold. I just want to be conservative. I say 200. It's really, oftentimes it's more than 200-fold enrichment. And the target recovery is consistently about 50%. So it works pretty well in terms of enriching those large fragments. All right. So in collaboration with Henley G at Stanford, recently we've developed a HRS catch plus 10x genomics workflow for targeted sequencing. So after you collect the catch product, and you don't have to do much at all. You just directly use that product, no cleanup is needed, and just load that to 10x Chromium box. And you then, for the library prep, then you go for Illumina sequencing. At the end, you use a link technology. You do de novo assembly to really haplotyping them. To give you more specific examples, the first one I want is human BRCA1 target locus, which we designed specific guides to cut a 200 KB fragment surrounding the BRCA1 gene. And at the end, we were able to get more than 150-fold target coverage. And the fold enrichment by sequencing showed 36-fold. We want to more aggressive, So this shows the link reads really enable successful phasing. And I want to emphasize here is that 50% of the link reads actually span the full length of the 200 KB BRCA1 fragment. That means the DNA out of HRS was really large, full length. We went more aggressively for probably one of the most important locus in our genome, the 4 megabases MHC region. So we designed two sets of guide RNAs, two sets overlap with each other, and in each set there are 21 guide RNAs, 21 locations and targeting different regions by generating a 200 kb interval. At the end, we'll pull them together for 10x genomic sequencing. We were also able to really recover the entire MHC locus, and the targeted coverage is 80-fold. At the end, we were able to really put together a single phase block over the entire 4 megabases MHC locus. You may wonder, you are targeting one locus. Can you really do like a number of loci at one time? Yes, we can. And we further characterized 38 known structurally variable regions in the genome. And we designed the guides for those regions, and we did the same thing. And at the end, we were able to recover all of those known variations, including 29 large deletions and 9 inversions. We frequently get asked by customers, friends, colleagues, and can you guys sequence the catch products directly on long-read sequencing platforms? And let's do a simple math. If you're talking about 200 kb out of a 3 billion base pairs genome, and your DNA input is only 2.5 micrograms, how much DNA are you going to get? So the math is about 150 picograms. So can that 150 picograms of DNA survive library prep? Even if they do, can you really push those molecules to the pores in PacBio or Oxford Nano platforms? It's challenging, right? So what we saw first was to do whole genome modification on that target product. So we did whole genome modification using QIAGEN's kit, and we sequenced it. It wasn't too bad, but it was not ideal as expected. So a lot of variation across the entire genome, and the coverage is really ranged from 0 to 50-fold. And some of the regions we were not able to cover. That's kind of expected because the amplification process was never truly random. So we really tried, and we come up with a different approach. By using a carrier-seq, recently we did this. We put the targets along with carrier sequences. We sequenced on Minnow, and we were able to actually detect that fragment. And the coverage now is not super high yet, but we were able to cover the entire region using that teeny tiny amount of target DNA and to reveal a 4 to 15x coverage, and the average is about 11x. So to quickly sum up, we can produce high molecular DNA, and with the HRS system, we can deliver very efficient in vitro CRISPR-Cas9 target enrichment. And the HRS catch allows local de novo assembly and a complete phasing of complex genomic regions coupled with linked read technology. And we did the direct long-read sequencing on catch products, and it shows some promises. And we're actively working on that, try to get even better results. So with that, I want to thank you for your attention. I'll be happy to take any questions.
One question. Time for one question. Anybody?
In a situation where you actually just care about phasing the entire locus, was there any actual value in doing the separation of the long fragment at first? Because you could have just targeted guide RNAs to that specific region and then.
Well, you really have to, if you want to preserve all the information in any of the long region, the 10x genomics technology is that it's a partitioning system, right? You prepare those templates without breaking them first. So they go into those droplets. By that time, it's one template per droplet. So it's really long. Then you sort of break them down not break them down, you do amplification within that droplet. Then you amplify those pieces, along that long fragment. But you just cannot, you don't want to start with small pieces first. Then it's going to be harder to assemble them later on. I mean, the problem with the short read sequencing is just assembly, right? You have all of those repetitive sequences, all this stuff. How do you put them back together when you do the assembly? So.